Sight, Vision. Thursday, May 14, 2020. 8:13 p.m.
Sight, vision. Structure of the eye. Conjunctiva is first layer light hits. Cornea transparent thick sheet of tissue, anterior 1/6. Anterior chamber space filled with aqueous humor, which provides pressure to maintain shape of eyeball. Pupil is hole made by iris, which determines eye color. Lens bends the light so it goes to back of eyeball. Suspensory ligaments attach to a ciliary muscle. These two things together form the ciliary body, what secretes the aqueous humor. Posterior chamber is area behind the ciliary muscle, also filled with aqueous humor. Vitreous chamber filled with vitreous humor, jelly-like substance to provide pressure to eyeball. Retina is filled with photoreceptors. Macula special part of retina rich in cones. Fovea completely covered in cones, no rods. Choroid pigmented black in humans, a network of blood vessels. BC black all light is reflected. Sclera whites of the eye, thick fibrous tissue that covers posterior 5/6 of eyeball. Attachment point for muscles. Visual sensory information. Sensation requires light - neural impulse by a photoreceptor. What is light? Electromagnetic wave part of a large spectrum. EM spectrum contains everything from gamma rays to AM/FM waves. Visible light is in the middle. Violet 400 nanometers, red 700 nanometers. The sun is one of most common sources of light. Light enters pupil and goes to retina, which contains rods and cones. There are 120 million rods for night vision. Light comes in, goes through pupil, and hits rod. Normally rod is turned on, but when light hits turns off. When rod is off, it turns on a bipolar cell, which turns on a retinal ganglion cell, which goes into the optic nerve and enters the brain. There are 6 to 7 million cones. 3 types, red, green, blue. Almost all cones are centered in fovea. Phototransduction cascade, when light hits rods and cones. Retina is made off a bunch of diff cells, rods and cones. As soon as light is presented to him, he takes light and converts it to neural impulse. Normally turned on, but when light hits it's turned off. PTC is set of steps that turn it off. Inside rod are a lot of discs stacked on top of one another. A lot of proteins in the discs. One is rhodopsin, a multimeric protein with 7 discs, which contains a small molecule called retinal, 11-cis-retinal. When light hits, it can hit the retinal, and causes it to change conformation from bent to straight. When retinal changes shape, rhodopsin changes shape. That begins this cascade of events. There's a molecule in green called transducin made of 3 diff parts alpha, beta, gamma. Transducin breaks from rhodopsin, and alpha part comes to disc and binds to phosphodiesterase, PDE. PDE takes cGMP and converts it to regular GMP. Na+ channels allow Na+ ions to come in, but for this channel to open, need cGMP bound. As cGMP decreases, Na channels closes. As less Na+ enters the cell, rods hyperpolarize and turn off. Glutamate is no longer released, and no longer inhibits on bipolar cells, it's excitatory to off bipolar cells. So bipolar cells turn on. This activates retinal ganglion cell which sends signal to optic nerve to brain. Photoreceptors, rods and cones. A photoreceptor is a specialized nerve that can take light and convert to neural impulse. Inside rod are optic discs, which are large membrane-bound structures, thousands of them. In membrane of each optic disc are proteins that fire APs to the brain. Cones are also specialized nerves with same internal structure as rod. Rods contain rhodopsin, cones have similar protein photopsin. If light hits a rhodopsin, will trigger the phototransduction cascade. Same process happens in a cone. Differences. 120 m rods versus 6 million cones. Cones are concentrated in the fovea. Rods are 1000x more sensitive to light than cones. Better at detecting light, telling us whether light is present, i.e. BW vision. Cones are less sensitive but detect color, 60% red, 30% green, 10% blue. Rods have slow recovery time, cones have fast recovery time. Takes a while to adjust to dark, rods need to be reactivated. Photoreceptor distribution in retina. Where optic nerve connects to retina, blind spot, no cones or rods. Rods are found mostly in periphery. Cones are found throughout the fovea, and few in rest of eye. If we zoom in on fovea, no axons in way of light, so get higher resolution. If light hits periphery, light has to go through bundle of axons and some energy lost. So at fovea light hits cones directly. Visual field processing. How our brain makes sense of what we're looking at. Right side of body controlled by left side, vice versa. How does it work in vision? All right visual field goes to left side of brain, all left visual field goes to right side of brain. Feature detection and parallel processing. Color, cones, trichromatic theory of color vision, form, parvocellular pathway good at spatial resolution, but poor temporal, motion, magnocellular pathway, has high temporal resolution and poor spatial resolution, no color. Parallel processing, see all at same time, simultaneous processing of incoming stimuli that differs in quality.